The conclusion of this project was that basically I found the solution to the proportion of the areas in the circle. It is not an exact way, it depends on a graph, it was impossible for me to find a direct value of x just knowing the radius and the proportion of the areas. But it should be able to be done. So the thing is that you cannot get exact values, and that's what makes estimates work so hard to manage. Well, the approximation was found, and was fully completed, and the program that I did, well, it does not really work, but it would frankly require more time than 6 weeks to be able to finish that.
Thank you very much. Rolando that is able to give you in English also.
Do you guys have any questions?
Does anyone here have any questions to ask? No?
Did you guys hear?
Listo.
Muchas gracias. Okay, entonces sigan.
Okay, our next student is Rolando Gonzalez. He will present his bacterial transformation in the gene of GFP.
Yes, the Greek fluorescent protein. Well, as the doctor says, my name is Rolando Gonzalez. Are you listening me? Yes?
Yes, we hear you.
Okay.
Okay, this one's in English, so you guys...
I was working in the Center of Biotechnology from here, the Tec Monterrey, and my work consists of to make a culture of E. coli, the most known bacteria nowadays, and with the gene of a transformation with the gene of the green fluorescent protein, the GFP. This protein is a tool of gene expression marker that serves as a gene expression marker. This gene is attached to the plasmid of a bacteria, so it can be expressed and it can be identified with a normal microscope, because this emits green fluorescence when it's excited with a source of ultraviolet light. Next. Well, my hypothesis was so in this scheme we can see a general way of my hypothesis. We extract the DNA of a jellyfish called Acroria victoria, and this DNA is inserted into the culture of E. coli, and the expected result is that the E. coli can show us the protein, so it can be fluorescent and it can be identified with a simple microscope. Yes, next. The procedure was so simple. First, we get the plasmid with the gene for the protein, and the problem was how to insert it into the bacteria. So we use a technique known as electroporation. This consists in give an electric shock of 6 volts to the cell culture and to ambulance the cell membrane, allowing the entrance of the plasmid to the cell host. Next. The next part, to be sure if the plasmid entered correctly to the cell host, we make a digestion with enzymes and a yield of electrophoresis. First, we digest the bacteria, recovering only the plasmid with the gene, and then the plasmid is colored, I don't know, with enzymes, so we can see them in a gene of electrophoresis to calculate the size of the plasmid. This procedure is made with a special kit that we get from the company Wizard Plus that has the protocol and the equipment necessary. Next. Well, this is a photograph of the gene of electrophoresis that shows us the different plasmids coded with different enzymes, and so we can predict the size of the plasmid that is how we expected, from 3 to 4 kilobyte bases of pair bases of, you know, the
Elena,
yes, that base. So the hypothesis was proved because the bacteria in the culture accepted and replicated the plasmid with the gene, and well, the size of the DNA fragments was expected, so we can conclude that the bacteria accepted and replicated correctly the plasmid that we inserted with the electrophoration. And that's it.
Presentation was about traffic modeling. Here is logo. This logo is a simple programming language that is very graphic. Exactly. And for the traffic modelation thing, there are 3 approaches: microscopic, mesoscopic, and macroscopic. The microscopic model is based on the individual reactions of every agent, that in this case is a car or vehicle, considering the actions of the car behind it. And the space-time behavior for each glycol and interactions between them are described by these kinds of models. But there is a little problem with this, because it's hard to make estimation or prediction with this kind of model. Next. Mesoscopic model is also called a kinetic model, and it uses methods of mechanical statistics to define a function that expressing the probability that a certain agent is in a certain point. And next. And last, the macroscopic model are focused on traffic as a global set of variables. They have like a time and localization variable as independent variables, and they consider density, flow speed, speed variance, and traffic pressure. Well, my investigation was based on the Nash model. It has 4 rules that you can see in the slide. If the maximum speed is not reached, each car will accelerate when needed. If the speed will be now the minimum between B and the one calculating the rule 1. B is the space between the car and the car next to it. The rule 3 is a random stopping that will occur with a probability P, less than 1 obviously. And the rule 4 is that the position will be actualized with the new speed. For this model, I made 2 verifications. One of them was a user control semaphore, and the second one was a random semaphore with a probability of 0.01 of changing the light. First, the effects of the semaphore in the Nash model. When you have about 5 cars, the model stops almost instantly. About 20, it starts fluctuating. About 50, fluctuations make themselves a little more notorious. And about 85 cars in this model, the semaphore makes kind of no difference. Next, please. Well, conclusions are that the presence of a semaphore doesn't let the mean speed to reach the maximum speed, which is fast reaching the original Nash model. Fluctuations are created by random stopping and quantity of cars. And traffic waves are creating about 15 cars, causing oscillation zones, which I cannot show you because I haven't been another person. Oscillation zones tend to change their place because of random stopping or semaphore effect. And with the saturation of the road, the presence of a semaphore makes little or no difference. And